All right, hey everybody. This is the lab overview video for our next big lab. So it's a 4-week lab, and this is otherwise known as the taste receptor lab. So I'll try and keep this overview short and sweet, but I want to give you kind of a broad picture of what it is we're trying to do in lab, right? So the big goal in this lab, kind of conceptually, is to try and connect these two things here, genotype and phenotype. It's kind of two things we'll explore a little bit more as we go through the semester. But hopefully these are terms you at least have some kind of understanding of, right? So let's start with this phenotype here. What phenotype are we going to be investigating in this lab? As the name suggests, we're going to be studying taste, and we're specifically going to be studying the trait of bitter taste sensitivity. And we're not going to, we'll talk a lot about bitter taste sensitivity on the whole, but we're specifically going to be investigating sensitivity to a chemical compound called PTC, which is phenylthiocarbamide, for those of you that are interested in that one, right? So this is a synthetic chemical that produces a really strong bitter taste for some people, but absolutely no taste for other people. And that's a very interesting phenomenon. And it turns out that there's a genetic underpinning for that taste difference. And so that's going to bring us to this side of the equation here, the genotype. So the genotype that underlies this trait are genetic variations, i.e. SNPs, in a specific gene. And this is the TAS2R38 gene. So this gene codes for a bitter taste receptor. And these SNPs actually alter the function of this taste receptor so that some people are sensitive, some people are not, and there's also kind of a range in between, right? So that's kind of broadly, conceptually, what we're trying to do in this lab. So what I want to do now is focus a little bit more on these genetic variations, the SNPs in this gene, okay? All right, so if this right here, this is the TAS2R38 gene sequence. Okay. Now there are 3 SNPs in this gene that we're going to be investigating. Okay, the first one is right here at nucleotide 145. There's another SNP at nucleotide 785, and the last one is at nucleotide 886. All right. So each of these varies is basically just kind of a single base variation. Okay. So at 145, individuals either have a C or a G. Okay. At 785, individuals either have a C or a T. Okay? And at 886, individuals either have a G or an A. Okay? Okay, so there's that notation you should be familiar with. Either a C or a G at 145, C or a T at 785, a G or an A at 886. Okay? Now importantly, these are all within the coding region of the gene, right? So these actually have an effect on the amino acids that are being coded by the sequence. Okay, so if an individual has this C, right here, this ends up coding for a proline or a P. If they have a G here, that codes for a valine or a V, okay? At this SNP, an individual with the C, this ends up coding for an alanine or an A. Whereas this T ends up coding for a valine, a V. Okay, at this SNP, if an individual has the G, this codes for a valine, V. Whereas if they have the A in this case, it codes for an isoleucine, an I. Okay, and so these amino acid changes functionally change this receptor. Okay. So let's kind of talk about now how these SNPs kind of combine together, right? So they're going to be 2 what are known as haplotypes. 2, we'll kind of give this a qualifier, 2 main or 2 common haplotypes for this gene. Okay, and haplotypes is really just fancy science for combinations of SNPs. Okay, and I've color coded these to make it a little bit more clear. Okay, so haplotype number 1 are all the purple SNPs. So this would be C, C, G. I'll make that more clear there. Right, so that's one of the main haplotypes here. Okay, and this, because of the way that this codes for these amino acids, P, A, and V, is oftentimes called the PAV haplotype. Okay, so you'll see it referred to as both kind of throughout the lab, CCG, PAV. Now, importantly, this ends up coding for a fully functional receptor. So this is actually what we call, this combination defines the taster allele. Okay, and allele is a term we'll define a little bit later. Okay, so this is a taster allele, and so these are individuals. If this allele ends up coding for a protein that's sensitive to PTC. Okay, now the other haplotype number 2, are the other SNPs. In this case, it's G, T, and A. Okay, and again, because this alters the coding region, sometimes this is known as the, this should be, mess this one up. This should be an alanine, right, A, okay, because this down here is what's known as the AVI haplotype. My mistake on that one, right? So you get the PAV and the AVI. So GTA, this second haplotype, codes for AVI, okay? Now this is what this ends up coding for, or forming the non-taster allele. Okay, so this ends up coding for a protein that's insensitive to PTC, okay? Essentially, this combination of SNPs produces a loss of function. Okay, so this is just a receptor that no longer transmits the sort of information when PTC binds to it, okay? So these are the 2 genetic variations that we're going to be investigating, okay? So with that said then, what is our goal here, okay? Our goal, or rather our goals, are kind of twofold, okay? Number 1, our goal is to determine your TAS2R38 genotype. Okay, that's our kind of primary goal. Now, along with that, what we can do is we can start to compare this genotype with your phenotype. We'll have a couple different methods we can use to assess your bitter taste sensitivity, both broadly and specifically to PTC. So we can also then explore consistency between genotype and phenotype. Okay? There are some interesting examples of where people's genotype and phenotype are kind of inconsistent. So there's some fun stuff to explore with this one, right? But again, this is our kind of primary goal right here, okay? Determining our TS2R38 genotypes. We're going to focus on that for the rest of this video. Okay, and we're going to end up using 2 methods for this one. Okay, and so right now I'm going to kind of work through those 2 different methods, just give a broad overview. We'll get into these in more detail when we get into lab, but this way you kind of have an idea of what to expect. Okay, so genotype method number 1 is what's known as the D-CAPS method. Okay, I'll go over that abbreviation when we get into lab right now. It's not really important right now, okay? But essentially what this does is it uses a combination of PCR and restriction enzyme digests to identify your genotype. So this roughly works like this, okay? So again, if this is our TS2R38 gene here, okay, and here we got our C145G, remind myself, C785T, and our G886A. Okay, this method focuses primarily, or really only, on this SNP right here. Okay, and that's because this SNP actually is sort of most associated with the taste preference. So if any of these 3 SNPs kind of appear to be kind of the most important for determining the function of the receptor, it appears to be this one. It doesn't mean that these aren't important, but this one seems to kind of take the cake a little bit, right? So what this method does, it uses PCR, or polymerase chain reaction. Okay, that'll be part of your lab intro for this one, to generate a product from right about here to here. Okay, this is the PCR product it generates. Okay, this is a product that is 221 base pairs long. Okay, so it includes that SNP right there. Now the other thing that this PCR reaction does is it actually introduces a mutation just upstream of the SNP. Okay, so this is a mutation introduced during PCR. Okay, The reason we do that is this is going to allow us to selectively create a restriction enzyme site in just one of the possible PCR products, right? So if you imagine here, there are two possible PCR products. If someone has a C here, you could get a PCR product that looks like this, okay, with the C, and then also with the mutation. Okay, remember this C defines the taster allele, okay? Whereas it's possible to have another PCR product if there's a non-taster allele in there that will have the same length, okay? It'll have the SNP there, but in this case it'll be a G, and there will also be that mutation introduced, okay? This is the PCR product derived from the lowercase non-taster. So this is the taster and non-taster. Okay? Now, importantly, this mutation only introduces a restriction site in this PCR product. Okay, so this mutation, what it does is it creates a restriction enzyme site, okay, an RE site. And this will be the basis for how we actually differentiate between the taster and the non-taster allele. So this is the rough overview. We'll do the PCR. We'll set up the restriction enzyme digest of the PCR. We'll run that out of the gel, and then we'll be able to tell whether or not the PCR came from this allele or this allele. Okay, that's our first method. Remember, that one focuses just on this SNP. Okay? We're going to use another method. Okay? And this one I'm kind of excited about. Okay? The second method we're going to use is DNA sequencing. Okay, we've been talking a lot about this in lecture, and so we actually have a chance to do some Sanger sequencing in lab. Okay? Now, as you can imagine, one of the drawbacks of the DCAPS method, the one we just talked about, is it really just identifies this one, right? And although there are two main haplotypes, there are kind of less common haplotypes that aren't just sort of sets of either this, this, and this, or this, this, and this. Okay, and so the DNA sequencing does a couple things. Number one, allows us to conclusively identify all three SNPs in this open reading frame. Okay, and it also allows us the possibility for picking up some of those more rare or less common haplotypes. Okay, so the way this is going to work is we're still going to start this with PCR. Okay, but in this case, we're going to amplify the entire open reading frame. Okay, so this is the TAS2R38 orth. Okay, going from a start to a stop. Now this will include all three of these SNPs in the PCR product. And then after a cleanup step, what we're going to do is we're going to take this PCR product and submit this for DNA sequencing. Okay, now if this works and we get some sequencing data back, we can then analyze this DNA sequence, and we should be able to conclusively identify all three of these SNPs. Okay? Now the way we're going to do the DNA sequencing is we're actually going to set up we're going to submit your sample for two different reactions. Okay? You're going to have one sequencing reaction with a primer going this direction. Okay? You're also going to have a primer going the other way. So in a lot of sense, this will look a lot, this is kind of like the paired end sequencing that they did in the Human Genome Project. So you'll generate a sequencing reaction going this way, okay, and one going this way. And really between the combination of these two DNA sequencing reactions, we should be able to identify all three of these SNPs, okay? That's the overview. Yeah, pre-lab is due before lab starts this week. It should be a fun lab. It's 4 weeks. We'll be kind of switching back and forth between this DCAPS method and the sequencing method, so really kind of do your best to really wrap your heads around each of those two things. As we move through the 4 weeks, I'll do my best to make sure we're all kind of on the same page. Should be a fun lab. Looking forward to it. Thanks.